After studying this module, you shall be able to know that the nucleotides are monomers which frame the DNA and the RNA. Next, we are going to learn about that nucleotides comprise of three components: pentose sugar, second being the nitrogenous bases, and 1 to 3 phosphate moieties. Next, we are going to understand that polynucleotides, whether it is DNA or RNA, are elements of heredity. We are going to know about nucleotides provide energy for many cellular processes. We are going to learn about the monomers as well as the polymer. Nucleotides and polynucleotides are biological molecules which play numerous roles inside the cell. Nucleotides, which forms the monomer, are the repeating units that form the polynucleotides, which forms the polymer. Polynucleotides are of two kinds: DNA, which is deoxyribonucleic acid, or RNA, which is ribonucleic acid. Just like we have letters in a sentence, the sequence of nucleotides in a polynucleotide carries the information. These are the biomolecules that carry genetic information. Nucleotides are essentially composed of three components: first, nitrogenous bases; second, a 5-carbon sugar; third, it is 1 to 3 phosphate moieties. DNA, which as we know is deoxyribonucleic acid, is much stable than RNA, but both are used to store genetic information by organisms. Few viruses use RNA as a genetic material. Now let's look at the three components of nucleotides. The first, let's study about nitrogenous bases. Nucleotides are comprised of single-ringed or two-ringed nitrogenous base. One-ringed nitrogenous base is called the pyrimidine, and two-ringed nitrogenous base is called a purine. Now let's look at the basic structures of pyrimidine and purine. Pyrimidine essentially is a 6-membered ring in which first and third are nitrogen atoms and 4 are carbon atoms. Now this is labeled in a anti-clockwise direction. Now if we look at the purine structure, it comprises of a pyrimidine ring fused to a 5-membered imidazole ring. Now let's learn about pyrimidines and purines individually. The pyrimidine bases are of three kinds. I generally remember it as cut, the C representing cytosine, U representing uracil, and T representing thymine. Cytosine and thymine are found in DNA, while cytosine and uracil are found in RNA. Now if we just look at their structures, we remember the original structure of pyrimidine ring. Now if we have cytosine will be labeled as 2-oxy 4-amino pyrimidine, while uracil is 2-oxy 4-oxy pyrimidine, and thymine being 5-methyl uracil. This was about the pyrimidines. Now let's learn about two-membered components of DNA or RNA, which are the purines. Now two kinds of purines exist in nucleotides: adenine, which is labeled as A, or guanine, which is labeled as G. Both are found in DNA as well as RNA. Now this adenine is basically 6 amino attached to the purine parent backbone, and guanine is 2 amino 6-oxy purine. Apart from A and G, there are other purines which exist in the cell but not as a part of polynucleotides. They are hypoxanthine, xanthine and uric acid. Hypoxanthine contains one oxomoiety which differs from xanthine which contains two oxomoiety, while uric acid is the most oxidized form comprising of 3 oxomoieties. Now let's learn about the inherent property of these nitrogenous bases. Whether it is purines or pyrimidines, they can undergo keto-enol tautomerism. These bases can exist in predominant keto or lactam form and rare enol or lactam form. These nitrogenous bases, because of aromatic ring structure, can absorb ultraviolet light. This characteristic absorption of UV light at 260 nanometer is useful in quantitative and qualitative analysis of bases, nucleotides and nucleic acids. We just now learnt about the nitrogenous bases, which are purines and pyrimidines. Let's move on to the next component of the nucleotides, which are the pentose sugars. Now these pentoses are the 5 carbon sugars present in polynucleotides. RNA contains D-ribose while DNA contains 2' deoxy D-ribose. The hydroxyl group at the 2' position in D-ribose is basically absent or not present in DNA. Rather, a hydrogen atom is present there. We shall see in later sections how this minor difference has far-reaching consequences on their properties. Nucleosides, which are formed by joining a sugar to a nitrogenous base via a beta-N glycosidic linkage. If the nitrogenous base is a purine, the nitrogenous base is linked to a sugar via its N9 atom, while if it's a pyrimidine, it is linked via its N1 atom. Nucleoside with purine as base are suffixed with ocene. Let's look at the nitrogenous bases. We are already acquainted with these particular structures. This is adenine, guanine, thymine, uracil and cytosine. When we move on to forming the ribonucleosides, to these bases we are going to attach a pentose sugar. In ribonucleosides, it is going to be a D-ribose sugar. Now in adenosine, it is the adenine moiety which is attached to a pentose sugar via the N9 atom. In guanosine, again the way we saw it in adenosine, it is attached to N9 atom to a D-ribose sugar. Next is 5-methyluridine. 5-methyluridine is a pyrimidine in which D-ribose will be attached to a N1 atom. Similar holds true for uridine and cytidine. Deoxyribonucleosides, just the difference which it contains from ribonucleosides is that the sugar which is present is 2' deoxy D-ribose. The atoms to which it is attached are N9 or N1 atoms of the pyrimidines. We learnt about the base hypoxanthine. When it is bonded to a ribose, it forms the nucleoside inosine. Now, nucleosides can exist in 2 conformations, either anti or syn conformation, which depends on the rotation of beta-N glycosidic linkage. For example, guanosine can exist in anti and syn forms. In syn conformation, the NH2 substituent which is there lies above the furanose ring, while in the anti conformation it lies away from the furanose ring. Now we just now learnt about the nitrogenous bases, the nucleosides, next let's learn about the nucleotides. Now these nucleotides are phosphates of nucleosides. Phosphoric acid can be esterified to sugar OH group via phosphoester bond. Ribose presents 3 such OH or hydroxyl groups which are essentially present at 2' position, 3' or the 5' position. While deoxyribose, because it lacks 2' OH group, has only 2 hydroxyl groups present at 3' or the 5' positions. Nucleosides, 5' phosphates, are predominantly found in the cell. If one phosphate moiety is attached to the nucleoside, it is called nucleoside monophosphate or NMP. Similarly, nucleoside di and triphosphates NDP and NTP are also found in the cell. Now let's look at this figure. We can see the glycosidic linkage linking the base to the pentose sugar, and there are 3 such hydroxyls in ribose moiety which is present at 2' position, 3' or the 5' position. Any of these hydroxyls can be phosphorylated. In this particular figure, they are phosphorylated at the 5' position. While we just now learnt about the ribose moiety. If we talk about that the sugar or the pentose sugar which is present is deoxyribose, it is going to contain only 2 hydroxyls at 3' and 5' position. The hydroxyl which is present at 2' position is absent in case of DNA. Now nucleoside monophosphate comprises of 1 phosphate moiety linked to 5' hydroxyl. Nucleoside diphosphate comprises of 2 phosphates linked to the 5' hydroxyl, and in case 3 are linked one in succession to another, it is known as nucleoside triphosphate. In this particular case, it will be called as nucleoside 5' mono, di or triphosphate. Now in the next section, we are going to look about the importance of nucleosides and nucleotides in the cell. Let's learn about adenosine first. Nucleosides serve as precursors to nucleotides, but adenosine, apart from serving as a precursor, also acts as a local hormone that circulates in the bloodstream and performs function like blood vessel dilation, smooth muscle contraction, and even regulates sleep. The nucleotides can even be cyclized. Nucleoside monophosphates can form intramolecular phosphoester bond, where the phosphoric acid moiety can be linked to 2 hydroxyls of the same ribose moiety, thus leading to formation of cyclic nucleotides. Now we learnt about the structure of ribose and deoxyribose. It has hydroxyls which are present at 3' and 5' position. Suppose a bond is formed, a phosphoester bond is being formed between the hydroxyls which is present at 3' and 5' of the same molecule. It will be called as a cyclic nucleotide in that particular case. 3', 5' cyclic AMP is one of the examples. 3', 5' cyclic GMP are the cyclic nucleotides that have important role in cellular signaling. They basically serve as second messengers. This is how a structure of cyclic AMP looks like. The base being the adenine, which is 6-aminopurine. The sugar moiety, as we can recognize from the 2' position, is the ribose moiety, D-ribose moiety. And the phosphate is linking the 3' OH and the 5' OH. This is what is known as a cyclic nucleotide. We are going to call it a nucleotide because it is containing a phosphate moiety as well. Now moving on to the other section, energy source for the cell. These nucleotides even serve as the energy source for the cell. Nucleoside 5' phosphates are prime source of chemical energy for the cell. ATP is called the energy currency of the cell. GTP is being given the job for protein synthesis in the cell, while CTP and UTP provide energy for phospholipid and carbohydrate synthesis respectively. These nucleotides even serve as building blocks for DNA and RNA. DNA and RNA are polymers of nucleotides. Two polynucleotide chains join basically in antiparallel fashion to frame the structure of DNA, while the RNA is only a single polynucleotide chain. Polynucleotides, let's learn about nucleic acids. Nucleic acids are biopolymers of nucleotides linked to each other via a 3' to 5' phosphodiester bond. This intermolecular linkage links 3' hydroxyl of one nucleotide molecule to 5' hydroxyl of another molecule, linking the adjacent nucleotides. Now remember, in cyclic nucleotides we learnt about that the 3' and 5' hydroxyls of the same molecule were forming the phospodiester bond. In this particular case, it is 3' from one molecule and 5' from the other molecule. Now, if the repeating unit is ribonucleoside monophosphate, it forms the ribonucleic acid or RNA. However, if the repeating unit is deoxyribonucleoside monophosphate, it forms the DNA or the deoxyribonucleic acid. One polynucleotide chain makes the RNA, hence it is single-stranded. Two polynucleotide chains make the DNA, hence it is double-stranded. Polynucleotide chains are represented in the figure, basically illustrates polynucleotide chain where adjacent nucleotides are linked by phosphodiester linkage. Now as we can see in this particular figure that this is the 3' moiety and this is the 5' moiety. So 5' hydroxyl is linked to 3' hydroxyl to form the phosphodiester bond. When such repeating units join one after the other, it is known as the phosphate deoxyribose backbone. In case it is a ribose sugar, it will be called as phosphate ribose backbone. Now from these, the bases project outward from this particular backbone. Figure shows the shorthand notation of our phosphodiester bond. In this particular case, the straight line is basically showing the 5 carbons of a pentose sugar. The slant line is going to depict the 3' to 5' phosphodiester bond linking the 3' of one pentose sugar to the 5' of the other pentose sugar, linking the two nucleotides. This notation can be used for DNA or for RNA. In this particular figure, we can see that this is essentially how a polynucleotide chain is represented. The first is this nucleotide comprises of adenine, a pentose sugar, a phosphate moiety whose 3' OH is attached to 5' OH of cytosine nucleotide via a phosphodiester linkage. And similar holds true for C is linked to T and then to G. This is a representation of a phosphodiester linkage. So hence we just saw a depiction of a sugar phosphate backbone with emphasis on depiction of 3' to 5' phosphodiester linkage. Let's summarize what we just learnt. Nucleotides are the monomers that make the polymer DNA or RNA. Nucleotides are comprised of 3 components: nitrogenous base, it can be a 6-membered ring which is a pyrimidine or a 9-membered ring which is a purine, a pentose sugar and a phosphate moiety. Nitrogenous bases can be 2-ringed purines or single-ringed pyrimidines. Pentoses are ribose or 2' deoxyribose. Nucleotide is nucleoside mono or triphosphate. Nucleosides and nucleotides perform many important functions in the cell. and DNA and RNA are polynucleotides.